Okay, we'll get started. So this is the last lecture for TRACO for 2014. And I'd like to thank Anthony Thomas over in Building 50 for the video cast, and Chris Graham up at Frederick for helping with the video cast. I'd like to thank Jonathan Wiest for supporting the TRACO class. And our first speaker today is Marina Dobrovolskaia. She's at the Nanotechnology Characterization Lab in Frederick. Her title, Immunological Properties of Engineered Nanoparticles and Some Challenges in Their Preclinical Characterization. Marina.
Thank you, Dr. Moody, for the introduction, and good afternoon, everyone. Good afternoon to those who joined us in this room, and also hi to everybody who is watching this presentation online. I will talk about immunological properties of nanomaterials, and in the beginning, I will start from just overviewing the definitions. We'll talk about what nanotechnology is, what nanomedicine is. We'll talk about examples of nanoparticle use in daily life. We'll see what nanoparticles can offer for cancer diagnosis and therapy, because this is of special interest to this lecture series. I will give you overview of nanomedicines. I will tell you about the lab which I am representing here. This is Nanotechnology Characterization Lab, or NCL. And then we will talk about immunological properties of nanomaterials. We'll see examples in the case studies of how nanoparticle physicochemical properties determine their interaction with components of the immune system. And I will also review some case studies to show you the challenges with preclinical characterization of these materials. So what is nano? There is no universal definition. The one which is most commonly used comes from National Nanotechnology Initiative, or NNI, and it defines nanoparticles as objects with at least one dimension at approximately 1 to 100 nanometers. However, the nanoparticles are used for biomedical application. The size can extend beyond 100 nanometers. And United States Food and Drug Administration, because of these purposes, specifically defines that if material properties are determined by the size of these materials, and the size is extended beyond 100 nanometers, it will still be considered as nanotechnology, and the size range extends to 1,000 nanometers. So we are talking about the objects in this size range, between the size of the molecule of glucose to approximately the size of bacterial cells. There are a lot of nanoparticles in daily life. According to the literature survey, there is about 800 manufacturer-defined products from more than 400 companies in more than 20 countries worldwide that use nanoparticles in their products. Nanoparticles are included in clothing, wound dressing, washing machine liners. They're used as a lens coating in sunglasses, in sporting equipment. Many of you are using nanoparticles throughout the summer. Then we apply sunscreens to our skin and the skin of our children, because some of these sunscreens contain nano-sized titanium oxide and zinc oxide. Nanoliposomes and emulsions are commonly used in cosmetics, and some materials like carbon nanotubes are used as structural materials. Nanotechnology is very special for cancer therapy, because, as you know, a lot of drugs are insoluble, and nanoparticles may act as carriers for hydrophobic drugs and serve to improve their solubility and also to achieve targeted delivery to the tissue where the drug is needed. Nanoparticles can offer multifunctional capabilities. Dr. McNeil, is director of Nanotechnology Characterization Lab, published a review back in 2005 where he described this concept of a nanoparticle drug carrier. This is a nanoparticle that has a nano-sized core, and It's representative of the multifunctional modalities because it contains image contrast agent, targeting molecule, and some drugs on the, incorporated into the particle. The nanoparticles are used not only for drug delivery, but also for cancer diagnosis as imaging agents, as diagnostic agents. And nanotechnology-based methods for detecting cancer cells are more sensitive. They work with smaller volume and provide greater sensitivity and specificity than conventional methods. Then it comes to cancer therapy. Nanoparticles efficacy occurs through two major mechanisms. One mechanism is considered as passive targeting, which relies on so-called enhanced permeability and retention effect, or EPR effect. So what this effect is based on is on leaky vasculature of the blood vessels surrounding the tumor cells. And nanoparticles, due to their smaller size, can penetrate this leaky vasculature, enter into the interstitial space surrounding the tumor where they release the drug. The second targeting mechanism is active targeting, and it is based on attachment to the particle surface of specific ligands, such as antibodies or small molecules or macromolecules like cytokines, which recognize their cognate receptors on the surface of cancer cells. However, it is important to mention that in order for the active targeting to work, passive targeting still has to take place, because even though we have antibodies or any ligand on the particle surface, in order for this ligand to recognize a receptor or molecule expressed on the surface of the cancer cells, nanoparticle has to go through the vasculature, get into the interstitial space, and then interact with cancer cell over there. So what is nanomedicine? Nanomedicine is a terminology which is commonly used by researchers to define nanoparticles that are used for biomedical applications. What I have on this slide is a summary demonstrating a difference between nanotechnology-based medical devices and drugs in investigational stage and then in the commercial stage. And as you see, among the nanomedicines that are currently under investigation, majority of the concepts are represented by medical devices, and only a smaller percentage is represented by the drugs. While if we look at the commercialized nanomaterials, majority of them are drugs, and some proportion, about 33%, is represented by medical devices. I prepared these graphs based on this publication by Etheridge et al. In Nanomedicine journal. If you're interested, I highly encourage you to read this article. It's very interesting. Two my other slides are based on the data published in this article. So now if we look at the nanomedicines based on the type of nanomaterials used to drug delivery, we also see the difference between investigational and commercial nanomaterials. In the investigational stage, the dominant nanomedicines, by dominant I mean representing more than 10% of the total concepts, is dominated by liposomes, followed by polymeric nanoparticles, followed by solid nanoparticles. And in the commercial stage, this proportion is different. In the commercial stage, there are more solid nanoparticles, followed by nanocomposites, and then followed by other categories. Actually, to me, this was very interesting difference, because I thought that majority of the nanoformulations that are approved for clinical use will be represented by liposomes and nanoemulsions. But as you see from this data, this is not the case. So what are the common features of these nanomedicines? If you look at this graph, you see that majority of nanomedicines are developed for cancer therapy, followed by infectious diseases and other applications. These concepts are designed for intravenous administration, and majority of them have size less than 350 nanometers. Most of these concepts are neutral, have hydrophilic surfaces. The reason for this is because neutral and hydrophilic surfaces are the most biocompatible and less toxic, and most of them are spherical in shape. This slide shows you the funding that National Nanotechnology Initiative spends and invests in the nanotechnology research. And as you see, even though in the last few years the budget flattened, it almost tripled compared to the 2001, then investment in the nanotechnology and especially nanotechnology for medical applications started in this country. So in drug development, there is such a concept as a valley of death. And I borrowed this image from a review by Butler in Nature that I think demonstrates this concept very nicely. So most of us who develop basic research concept for diagnosis or treatment of cancer are on this bank in drug discovery. And in order to transition this wonderful concept into the clinical use, before patients see the benefits of this concept, there is a huge gap. And this gap is called valley of death because crossing this valley is not fast and it's not easy. And it requires efforts from both academia and industry. And for emerging technologies such as nanotechnology, there is a wonderful place for the government to help this transition. And one of such efforts is the Nanotechnology Characterization Lab, or NCL. Our lab was established back in 2004 as interagency, under interagency agreement between National Cancer Institute, National Institutes of Standard and Technology, and United States Food and Drug Administration. And laboratory was tasked with preclinical testing of nanotech drugs and diagnostics. So what NCL is doing, we're performing preclinical work, helping academia, helping small biotech companies and big pharma to cross this valley of death for nanomedicines. We do so by conducting comprehensive assay cascade, which includes physicochemical characterization, and then in vitro and in vivo toxicology, pharmacology, and immunology. From the very beginning, in 2004, the lab is led by Dr. Scott McNeil, whose picture you see on this slide. We've been in operations for 10 years. In fact, in September this year, we celebrated our 10th anniversary. And during this time, we characterized more than 300 different nanoparticle concepts. What you see on this graph is the different kind of particles that passed through NCL assay cascade. You see that the dominant particles are liposomes, followed by polymers, metal oxides, and metals. We also worked with dendrimers, emulsions, nanorods, quantum dots, fullerenes, micelles, carbon nanotubes, nanocrystals, and many other formulations. Later in my presentation, then I will show you case studies. I will show you our experience with some of these materials. It is very well established now that nanoparticle physicochemical properties such as particle charge, size, hydrophobicity, and the presence of targeting moieties that determine the interaction with biological systems. My expertise is in immunology and immunological characterization, so majority of the case studies demonstrating this point will be used from our immunological characterization. Nanoparticles can be immunotoxic. However, this immunotoxicity can be good or bad, depending on whether it is desirable or not. A lot of engineered nanomaterials are designed for biomedical use, and they are designed to be immunologically reactive. Nanoparticles can be used to increase vaccine efficacy, to improve cancer immunotherapy, to achieve therapy of inflammatory disorders or autoimmune diseases and infectious diseases. So these kind of interactions with the immune system are good because they are desirable. However, nanoparticles may also lead to undesirable side effects, including hypersensitivity reactions, anaphylaxis, coagulation disorders, myelosuppression, and when it is undesirable, may lead to lower body defense to pathogens and cancer. And many of these examples come from the accidental or environmental nanomaterials. We at NCL are focused on the engineered nanomaterials that are being used for diagnosis and therapy of cancer. So they are not necessarily intended to be immunologically reactive. And what we want to know whether these materials have undesirable effects. Why? Because once we understand the immunological reactivities and the particle properties that are responsible for these immunological reactivities, we may inform drug development process and develop these concepts to reduce these toxicities. Nanoparticles can be immunotoxic. However, so far there is no new immunotoxicity which was reported for nanomaterials. When I'm saying new, I mean immunotoxicity which would be related to nanoparticle size. Nanotechnology-based pharmaceuticals are not more intrinsically immunotoxic than other drugs in current clinical use. Moreover, there is increasing number of data demonstrated that incorporation of the small molecule or macromolecular drugs onto the nanotechnology platform helps decreasing immunotoxicity of these drugs. And what I will do now, I will give you two examples, one from the field of the small molecules and one from the field of the therapeutic proteins. Many of you are familiar with anti-cancer drug doxorubicin. When this drug is used in clinic, many patients develop so-called disseminated intravascular coagulation, or DIC. This disorder originates from development of so-called procoagulant activity, or PCA complex, on the surface of the leukocytes. And what this complex is doing, it triggers plasma coagulation cascade, leading to the blood clotting. Many patients have to be withdrawn from the therapy because of these coagulation side effects. Incorporation of this drug into the pegylated liposome is known as doxil, and this formulation does not have this type of toxicity. Another example is therapeutic protein tumor necrosis factor alpha. This cytokine, the name of this cytokine stands for its function, right? So it's a factor that causes necrosis of the tumors. But in order to achieve therapeutic efficacy of this protein, one has to deliver so much protein that it is incompatible with the patient's life, because TNF is very potent immunostimulant. There is a small biotech company here in Rockville, Maryland, called Site Immune Sciences. So what this company did, they put TNF on the surface of colloidal gold nanoparticles coated with polyethylene glycol. So what polyethylene glycol is doing, it makes particle surface hydrophilic, and it shields TNF from immune cells recognition, so that particles stay in the circulation while it is delivered to the tumor site. And during this circulation time, PEG shields TNF from immune cell recognition. That decreases the systemic immunostimulation and allows administered in higher concentrations of the TNF to achieve therapeutic efficacy from this drug. And this concept is currently in clinical trials stage. There are a lot of challenges in characterization of nanomaterials. Back in 1968, Kiuru Ishikawa suggested using what he called cause-effect diagram or fishbone diagram to summarize potential factors that may have overall effects. These days, this diagram is known as Ishikawa diagram, and it is used in industry, mostly in the product development and operation management. But I use this idea to prepare this fishbone diagram to summarize factors that are critical in chemistry, efficacy, pharmacology, toxicology, and hematology and immunology of engineered nanomaterials. The knowledge that I used to prepare this slide is generated over many years of working side by side with two of my colleagues at NCL, Dr. Anil Patri, who for many years, for almost 10 years, led NCL chemistry group, and Dr. Steve Stern is currently head of pharmacology and toxicology section. So what you see on this slide are the factors that are critical to understand while transitioning nanotechnology concepts from bench to bedside. In chemistry, they include particle size, charge, shape, composition, surface coating, surface ligand density, solubility, stability, architecture, and excipients. For efficacy, it is important to use models that are appropriate for the certain type of nanoparticles. Drug loading is critical, drug stability is critical, drug release, as well as targeting. In pharmacology and toxicology, Dr. Stern summarized these factors, including the biodistribution of nanoparticles, again, the use of appropriate models to study toxicity, long-term chronic toxicity, mechanisms of toxicity, predictable in vitro methods. There are a lot of bioanalytical challenges, and also excipients that are included in nanoparticle formulation sometimes cause a problem for toxicology. This is my area of expertise. Based on our experience with hematological, immunological characterization, I can say that very challenging and a lot of problems come from chemical impurities in nanoparticles and biological impurities such as endotoxin. And later in my presentation, I will talk about this. Toxic excipients, sterilization, predictive in vitro methods, deparagenation, predictive in vivo models, endotoxin detection and quantification, and exaggeration of inflammatory properties of endotoxin by nanomaterials. And I think from just looking at this slide, you can appreciate that many of the factors are overlapping between the chemistry, toxicology, and immunology. So this is why I think the core strength of our lab is the three sections, the three groups working together. The chemistry, toxicology, and immunology are inseparable because I cannot evaluate immunological compatibility of nanoparticles without knowing their physicochemical properties. And later in my presentation, I will show you case studies demonstrating this connection. This slide is prepared by my colleague at NCL, Dr. Steve Stern, and the main idea of this slide to show that nanoparticle stability is very critical for to achieve very good drug delivery and good efficacy in terms of cancer therapy. If nanoparticles are too stable, these particles do not release the drug or release the drug very slowly. As a result, they are non-efficacious and they are accumulating in the tissues. And as example, we are showing here the concept which includes anti-cancer drug paclitaxel conjugated to a dendrimer nanocarrier. In this case, then we tested this concept in our toxicology and immunotoxicity assay cascade. We found no toxicity. It was perfect, non-toxic drug. However, what you see from this graph, it did not release the drug. Even less than 10% of the drug was released over 48 hours. As such, it was not toxic, but it was also not efficacious. On the another extreme point are unstable formulations. Unstable formulations are those that release drug too quickly, and that results in off-target toxicity and also not optimal efficacy. What we are seeing here is an example of such unstable formulation, which contains drug docetaxel on the liposome. What Steve found by studying this nanoparticle in vivo, that the drug is released from the liposomal carrier within first 5 hours, and it is causing off-target toxicity and is not optimally efficacious. Nanoparticles may change by distribution of the drug. Again, I'm using an example of the anti-cancer drug doxorubicin. You know that this drug accumulates in bone marrow, and this is responsible for the myelosuppression as a side effect, and also it distributes to the heart, and as a result, doxorubicin common toxicity is cardiac toxicity. However, then this drug is incorporated into the different nanoparticle platform. These particles may change distribution of the drug. In case of the doxil, which is pegylated liposomal doxorubicin, from bone marrow and heart, the drug moves into the skin. And so as a result, doxil has no myelosuppression and no cardiac toxicity issue. However, due to the accumulation in the skin, now it has new toxicity known as PPE or palmar-plantar erythrodysesthesia. Another example includes same drug incorporated into the cyanoacrylate nanoparticle. Again, instead of the bone marrow and heart, this nanoparticle takes doxorubicin into the kidney. And as such, there is no cardiotoxicity, there is no myelosuppression issues, but there is a nephrotoxicity. So it is very important to understand when we use nanoparticles as a drug carriers that change in the drug carrier may result in altered distribution, which may change toxicity profile of the drug. I mentioned to you when I was showing you the Ishikawa diagram that very often excipients, not nanoparticles, but excipients used to formulate the drugs into the nanoparticles are toxic. In this case, I'm showing you the drug-loaded nanocapsules. Then this concept was evaluated in my group. We looked at the compatibility with the erythrocytes, and we found that this nanoparticle formulation is hemolytic. Normally, we use 96-well plate assay to detect plasma-free hemoglobin, but in this case, we didn't even need that test. You can see that there was no single erythrocyte left. Everything was lysed. What is the reason for this toxicity? Then our chemistry group did physicochemical characterization, and when they performed dynamic light scattering analysis of these nanocapsules, they detected micelle with a size approximately 165 nanometer, which was expected theoretically for these particles. But then the same formulation was analyzed by transmission electron microscopy. In addition to the nanocapsules, they also detected micelles. And what we also found out by talking to the manufacturer of this particle, that this surfactant, Bridge 78, is used as a component in the synthesis process. Bridge 78, like many other surfactants, then it is present at the concentration above critical micellar concentration, or CMC, forms micelles. And so these micelles could be reproduced, and you see they look identical to what we observed with the formulation. But then concentration of surfactant is below its critical micellar concentration, these micelles don't form. These surfactants, they may interact with erythrocyte membranes, with cellular membranes, and cause toxicity. So the toxicity in this case is caused by excipient and not by nanoparticle. Particle size is important. What I would like to show you on this slide is the relationship between nanoparticle size for the cationic PAMAM dendrimers. The similar relationship exists for other types of the particles, but all particles are different, and I'm just using one example. We study here interaction of the particles with platelets, and we are using platelet aggregation as an endpoint. As we transition from the smaller particle to the larger particles, they reactivity with platelets membrane increases. So the particle size is bigger, the ability to induce platelet aggregation is greater. Again, particle size is important. However, size in biological context may be different, and this is due to the interaction with plasma proteins. Many of you may have heard this definition as protein corona, which refers to the plasma proteins bound to the particle surface after introduction of these particles into the bloodstream. This is from our early studies with citrate stabilized colloidal gold nanoparticles. These particles have theoretical size of 30 nanometers, and when these particles are studied under pristine conditions, it doesn't matter whether we take transmission electron microscopy, atomic force microscopy, or dynamic light scattering to measure their size. We measure the size which is very close to its nominal size of these particles. However, after these particles are incubated with human plasma, what we see is that size measured by TEM and AFM does not change, because these two methods look at the core part of the particle. But hydrodynamic size of the particles, as measured by dynamic light scattering, almost doubles. So what is important here is our body sees these 30 nanometer gold particles as 80 nanometer particles, not as 30 nanometer particles after the particles are introduced into the biological systems because of the protein corona formed on the particle surface. And this is very important to recognize. Particle surface charge is another property which is commonly used to evaluate their reactivity with biological systems. Again, in this case, I'm using example of the cationic PAMAM dendrimers. On the earlier slides, I showed you that cationic PAMAM dendrimers interact with platelet surface and induce platelet aggregation. What our chemists did, they masked 25% on amines on the particle surface, and we use these particles to look at the platelet aggregation. We saw decrease in reactivity. Then another 25% is blocked, and we see again decrease in platelet aggregation. And as we keep masking the amines on the particle surface and completely neutralize the surface, we completely neutralize reactivity of these particles with the biological membranes. So again, this case study demonstrates that zeta potential is important. The less the number of surface amines, the less their reactivity with platelets. Particle composition is important. In this case, I am comparing two different dendrimers, the PAMAM dendrimers or polyamidamine dendrimers and triazine dendrimers. If we look at the triazine dendrimers, you look at this blue graph, you see the same trend that we observed with PAMAM dendrimers, the red bars, in that if we transition from the smaller particle generation 3 to the larger particle generation 7, we see increased reactivity with the platelets. However, if we compare same generation triazine dendrimers with PAMAM dendrimers, we see that triazine dendrimers are less biologically reactive than their PAMAM counterparts. Why is that? The architecture of this is triazine dendrimers and this is PAMAM dendrimers. You see that their architecture is different. At the same generation, triazine dendrimers have less number of the surface groups on the particle surface, and we know from the previous studies that the number of the surface amines are important determinant of particle reactivity with biological membranes. So I hope that by now I convinced you that particle size and zeta potential are important determinants of nanoparticle toxicity. However, not always they allow to discriminate between nanomaterials displaying different biological effects. And as a case study, I would like again to use doxils, which is pegylated liposomal doxorubicin. There are many formulations of doxil available worldwide. That includes doxil, Lipodox, made by Indian company Sun Pharma, Calyx in Europe, Cyanodoxazome in Iran. In my case study, I will talk about these three formulations because those formulations are available in the United States. Then our chemistry group did analysis of the hydrodynamic size of pegylated liposome and also zeta potential. They did not detect any difference between what we called old doxil. This is a pegylated liposomal doxorubicin available in the United States before 2012. Then there was the company manufacturing these particles experienced some problems with in their facilities, which resulted in worldwide shortage of doxil. Then these problems were addressed at some point. The same manufacturer produced new doxil, and so this is a doxil available in the US after shortage, and we compare it to the Lipodox, which is pegylated liposomal doxorubicin manufactured by Indian company Sun Pharma. According to the physicochemical characterization, hydrodynamic size and zeta potential, there is no difference between these formulations. However, then we look at the complement activation, we do see the difference. And then the doxil, the old doxil, is reactive with complement. In fact, we used this material as a positive control in our complement activation studies. But the new doxil and the Lipodox do not have this toxicity. And this data are in agreement with in vivo clinical data and data in animal model of CARPA. CARPA stands for the complement activation related pseudoallergy, and this is very common dose-limiting toxicity of doxil. In this case, this is very good news for the patient in that whatever the difference in the new formulation, the toxicity was decreased, but this relationship could be the other way around. So this is why it is very important to understand what properties, in addition to hydrodynamic size and zeta potential, are responsible for the discrepancies in biological properties of these materials. Now I will switch the subject a little bit and talk about endotoxin. For those of you who are not familiar with endotoxin, I'll give very brief introduction. Endotoxin, or lipopolysaccharide, is a component of the membrane of gram-negative bacteria. And it is very potent immunostimulant. Even when it is present in picogram concentrations, immune cells detected and react to it by production of inflammatory stimuli such as cytokines. Endotoxin may lead to immunostimulation and also septic shock, which results in multiple organ failure. Nanoparticles can be contaminated with endotoxin during synthesis procedure. And endotoxin contamination is very common, especially for nanomaterials in preclinical stage. In fact, based on our experience at NCL, about 30% of preclinical nanoformulations fail due to the endotoxin contamination. Endotoxin in nanoparticles is bad because it results in generation of erroneous data that can give us wrong conclusion. It may confound results of efficacy and toxicity studies. It may be responsible for the undesirable toxicity. Also, because some nanoparticles, not all, but some nanoparticles may exaggerate inflammatory properties of endotoxin, and also because it may create problems with immunogenicity of protein-based active pharmaceutical ingredients or targeting ligands of nanoparticles. And what I'm showing you here is induction of procoagulant activity by various concentrations of LPS alone and in the presence of cationic dendrimers. Cationic dendrimer by itself does not induce PCA, but then combined with LPS, you see increase in the procoagulant activity. In preclinical characterization of nanoparticles, one of the biggest challenges, one of the grand challenges, in my opinion, is accurate detection of endotoxin. What I did here, I summarized some of the examples from NCL assay cascade, looking at the different nanoformulations and amount of endotoxin detected in this formulation by 3 different forms of the limulus amoebocyte lysate assay, or LAL assay, which is routinely used in pharmaceutical industry to quantify contaminating endotoxin. What we found is that majority of the nanoparticles interfere with the gel clot assay. For some nanomaterials, like this dendrimers, it doesn't matter whether we use gel clot, chromogenic LAL, or turbidity LAL. Results are very consistent. But for some materials, like in case of this nanocrystal formulation, relying on the gel clot may lead to the overdose of endotoxin. And the reason why this is important is because gel clot LAL is FDA preferred method. So what we did, we conducted collaboration with FDA, looking at the different nanoparticles and the ability of these particles to interfere with the gel clot and other LAL tests. And based on these studies, we proposed this decision tree, which informs us in choosing appropriate LAL format for the given nanoparticle formulation. This decision tree is based on physicochemical properties of the nanoparticles. If nanoparticles have absorbance at 4 or 5 nanometers, which is a wavelength of the chromogenic LAL assay, then we don't use chromogenic LAL for this formulation. We use turbidity and a gel clot. If nanoparticle formulation is turbid, then we don't use turbidity assay, then we use a gel clot and chromogenic assay. But the main message from this decision tree is that we use more than one LAL formats for the same nanoformulation, and at the end we compare the data. If results from 2 different LAL are in agreement, then we report LAL results, but if they are in disagreement, then we use biological assays such as rapid pyrogen test or macrophage activation test to verify LAL findings. This approach is very useful in many cases, and this is one of the examples. This nanoformulation was characterized in NCL assay cascade, and then we tested it by chromogenic LAL. We detected 0.5 EU per milliliter milligram of the nanoparticle in this formulation. The same formulation tested by the turbidity kinetic LAL gave us 21.4 EU per milliliter. So how do we know which of this data is correct? At the dose at which this material is used, if this value is true, then material should not be pyrogenic. But if this value is true, material will be pyrogenic. So this is when we used rabbit pyrogen test, and we found that this material is pyrogenic. At this point, we returned this formulation to the submitter, and we worked with them on purifying this formulation from endotoxin contamination. Then purified formulation, was tested by LAL assays again. You see that the results of the LALs are in agreement, and they detected much lower quantity of LPS, and also the material was non-pyrogenic in the rapid pyrogen test. So this case shows us the utility and the usefulness of the supplementing LAL test with the biological assays. But not all this approach is working. And this is an example where biological tests are not always helpful. This is commercial formulations and nanoformulations, Abelset, Abraxane, Visodyne, and Ferrahim, which was studied by turbidity, chromogenic, and gel clot LAL. And as you see, that some of them, like in case of Abraxane, there was no problem for different LALs, but for example, for this formulation, Visodyne, we do see discrepancy between turbidity, chromogenic, and the gel clot LAL. Please note that all of them are negative in terms of endotoxin by the gel clot assay. But when we use these particles to verify LAL finding with the microphage activation test, one of the important steps is to see the validity of the test. In this case, we take these particles and we deliberately spike them with endotoxin at the known concentrations. And we use this to see whether we can recover this endotoxin, which tells us whether the test provides us valid data. And we see that in case of the Abelset and Ferrahim, the data is reliable because we can recover endotoxin which was spiked into these samples. But in case of Abraxane and Visodyne, this approach is useless. We cannot detect any of the endotoxin because these materials contain cytotoxic drugs that kill the cells and inhibit endotoxin spike recovery. So still, there are some approaches to find out endotoxin contamination in nanomaterials, but it is not straightforward and it requires the use of combination of the methods. Another very important parameter in studying endotoxin contamination in nanomaterials is the use of various reagents. In immunology, we have a lot of the reagents that are known to neutralize endotoxin inflammatory properties. One of such reagents is polymyxin B. Polymyxin B is a polycationic peptide. It is antibiotic that is used to treat topical bacterial infections. And what immunologists commonly do, then they have a doubt whether the immunostimulation is caused by endotoxin or by test material, in our case nanoparticles, we use polymyxin B to add into the reaction mixture, and then we see whether we inhibit biological effect or not. For example, here we look at the cytokine induction by LPS, and by LPS in the presence of polymyxin B. We completely inhibit cytokine induction, and same true in this case. We study metallic nanoparticles, and in both cases, these metallic nanoparticles induce cytokines. Then this formulation is tested in the presence of polymyxin B. We do not see inhibition of the cytokine secretion, but in this case, we do see inhibition. So the traditionally trained immunologist would interpret this data as, in this case, cytokine induction is caused by nanoparticle, while in this case, this cytokine induction is caused by endotoxin contaminating nanoparticle. But this is not necessarily the case. And I would like to share with you some data showing that polymyxin B, because it is cationic material, may interact with the nanoparticle and inactivate nanoparticle as well. It is inactivating endotoxin because polymyxin B interacts with LPS electrostatically. So any anionic material can also be neutralized by polymyxin B. In this case, the metallic nanoparticles have hydrodynamic size of about 26 nanometers and approximately minus 47 millivolts of Zito potential. But in the presence of polymyxin B, particles aggregate to a micron size, a TLS is unable to measure the size of these particles, and you can see it by transmission electron microscopy. And the zeta potential of these materials is neutral. From minus 47, it becomes 9. So polymyxin B is a useful reagent sometimes, but it has to be used with caution in that it may change physicochemical properties of the nanoparticles as well. And this is one of the challenges in preclinical characterization, because in addition to characterization of nanoparticles under pristine condition and under the physiologically relevant conditions, we also have to understand whether additional reagents that we use in our biological tests can influence particle physicochemical properties. And if they alter particle physicochemical properties, that the results of this test may not be valid. Nanoparticle sterilization stability is another grand challenge. I'm using an example on this slide of the citrate stabilized gold colloids and citrate stabilized silver colloids. Gamma radiation is a very common method for the sterilization, and also it is a very common method for depyrogenation. When gold nanoparticles are sterilized by gamma radiation, their properties do not change, but silver nanoparticles disappear. And what we see appearance of these beautiful structures that look like ribbons and flowers and swans, but anything but the nanoparticles. In fact, the gold material, which is available from NIST as a standard reference material, is sterilized by gamma radiation. But in case of the silver, not only particle shape and size changes after gamma radiation, their biological properties also change. You see, this is unsterilized silver colloids, which are do not react with platelets, but as we keep increasing the dose of gamma radiation, they become more toxic and interact with platelet membrane, resulting in platelet aggregation. I would like to finish my presentation reviewing another grand challenge, which is not unique to nanotechnology, which is common for other areas of drug development. And this challenge is related to in vitro and in vivo correlation. The likelihood of identifying immunotoxicity or any other types of toxicity increases as we progress from preclinical in vitro to preclinical in vivo into clinical trials. But in vivo tests are more expensive and they have less throughput. Therefore, their application in preclinical studies is limited. Therefore, there is a growing recognition of the need for the rapid screening methods, and there is a requirement for those methods to be predictable and valid and give us informative and reliable data. Cascade of validated, reliable, and predictive methods can address this need, and there is an effort worldwide in understanding predictability of in vitro tests. I would like to step back here and give you an example of the lesson which was learned from biotherapeutics to demonstrate why in vitro methods, predictable in vitro methods, are important. I will talk about TGN-1412 compound. So what is it? What happened here? There was a company in Europe, Digenir Immunotherapeutics, that developed an antibody, supermonoclonal antibody, that recognized CD28 on the surface of the T cells. So what these antibodies was expected to do? For the T cells to interact with the antigen-presenting cells, two events are necessary. One is interaction between T cell receptor on the surface of the T cells with MHC peptide complex on the surface of antigen-presenting cell. This interaction gives information to T cells that there is an antigen T cell has to react to. But this interaction is not enough to stimulate activation of the T cells. For this, the second interaction is necessary between CD28 on the T cell and co-stimulatory molecules CD80 and 86 on the surface of the antigen-presenting cells. Then this second interaction takes place. T cells now has okay or green light to go with the proliferation, production of cytokine and activation in response to the antigen. But in cancer, for example, there is a mechanism that cancer cells develop to inhibit this interaction, which inhibits T cells. And the idea of this CD28 antibodies was to stimulate CD28 to promote T cell activation. Then this concept was studied in preclinical studies. The studies were done in non-human primates and in rodents. And there was no cytokine storm identified during these studies. However, then they transitioned into the clinical studies and injected this material into human volunteers. 6 out of 6 patients were delivered into the intensive care unit due to the cytokine storm. They almost lost their life. They had to stay in ICU for months, and some of them lost their fingers and toes as a result of the cytokine storm. What you see here is a necrosis of the tissues due to the overwhelming production of a tumor necrosis factor alpha. This was very devastating example, and the research community, especially in biotechnology proteins, was very affected by this. Later, then studies were done to understand the reason for this toxicity. We found that they found that in vitro experiments using peripheral blood mononuclear cells exposed to this compound showed high levels of TNF. So if this in vitro test was used to supplement preclinical studies in monkeys and rats, then the life of the patients may be saved, and maybe was not placed in such a danger like in this case. So this example demonstrated the necessity and importance of the in vitro tests. Luckily, nothing like this happened in nanomedicine, but in nanomedicine, we use cytokine tests to predict some of the nanoparticle toxicity due to the cytokine storm. What I show on this slide, I try to summarize the methods that are used in preclinical nanoparticle characterization to look at the immunocompatibility of these materials. We know from our work at NCL and also from the literature that the common acute toxicities for nanoparticles include hemolysis or damaging of erythrocytes, complement activation, thrombogenicity, phagocytosis, pyrogenicity, which is ability to induce fever, and cytokine induction. And luckily, most of this toxicity in vivo can also be detected using a battery of the in vitro tests. I would like just to show you one of the examples where in vitro methods are predictive and helpful in identifying these toxicities. In this case, we look at the metal oxide nanoparticles that have identical core, identical hydrodynamic size, and identical zeta potential, but the surface of these particles contain different ligands. And I just, for the purpose of this presentation, I named them nanoparticle formulation 1 and nanoparticle formulation 2. These particles were studied in preclinical studies in vivo, in rodents, and in rats and in rabbits. Endotoxin was undetectable in both formulations by gel clot LAL, and formulation 2, but not formulation 1, was toxic. Then our pathologists analyzed the tissues from these animals. They found congestion, which was very similar to that seen in septic shock. So even at some point, we thought that these particles are contaminated with endotoxin. But again, they did endotoxin tests and did not find any endotoxin. An analysis of the plasma samples from the animals showed high cytokine levels. Then these particles were tested in vitro in peripheral blood mononuclear cells. The toxicity was reproduced for the formulation 2. Formulation 2 was inducing TNF, IL-1, IL-8 cytokines, but formulation 1, which was not toxic in vivo, did not induce the cytokines. Again, this study shows the utility of the in vitro cytokine test in predicting in vivo toxicity due to the cytokine storm. So in summary, I would like to say that each nanoparticle is unique. Particle physicochemical properties are very important. Comprehensive characterization is very important, not just DLS and zeta potential. And the key factors that are critical in preclinical to understand during preclinical development of nanomedicines include physicochemical properties such as particle size, charge, composition. Their functional properties that include stability, drug loading, drug release, targeting, It is important to understand structure-activity relationship because the beauty of nanotechnology is that particle physical-chemical properties can be engineered. And by engineering these properties, we may deliver interaction with certain components of the immune system or certain components of the biological systems, or we may avoid these interactions. Endotoxins and sterility, as well as sterilization and deparagination of nanomaterials, are very common challenges during preclinical development, and the need for predictive in vitro methods like the cytokine induction test. With this, I would like to say thank you to my colleagues at Nanotechnology Characterization Lab. I would like to say that I am very proud for being a member of this group. Dr. Scott McNeil is laboratory director. Dr. Steve Stern is our head of pharmacology and toxicology section. We have very talented scientists with expertise in drug delivery, chemistry, immunology, oncology, cell biology, and we have wonderful technical support team. Majority of the data that I shared with you today in immunology assays are generated by Tim Potter, Barry Noon, and Jamie Rodriguez. I also would like to say thank you to NCL collaborators for providing their materials to us, and to our collaborators outside of the NCL. Dr. Jan Simak from US FDA CBER is a year-long collaborator whom we worked with studying interaction between nanoparticles and coagulation system. Dr. Eric Simanek from Texas Christian University provided triazine dendrimers so we could understand the importance of the particle composition and how particle composition influences biological reactivity of these materials. If you have any questions after this presentation, please feel free to send email. If you want to, you can contact me by email, and you can also visit NCL website to look at our publications, to look at how you can collaborate with NCL if you have a nanoparticle concept and you would like help transitioning this concept from clinic to the bedside. And with this, thank you very much for your attention, and we have a few minutes to answer questions.
Sorry if I missed this at the beginning, I came in a minute late. So how many nanomedicines are approved and being used in the clinic?
The question was how many nanomedicines are approved and being used in clinic. I cannot tell you like a number. I don't have a magic number. I just would like to say that there are a number of the nanomedicines currently in the clinical use. And for cancer therapy, two very common examples are Abraxane, which is anti-cancer drug paclitaxel bound on the nanoalbumin particles. And another one is Doxil, which is pegylated liposomal doxorubicin. As far as I know, well, nanoparticles are developed to target different types of cancers, but Abraxane is used for breast cancer. And the advantage of this medicine is not specifically to target the cancer cells, but to reduce toxicity, which is common to the traditional formulation of a paclitaxel. For example, in Taxol, paclitaxel is insoluble, and clinical formulation of the Taxol contains Cremaphore-AL. Cremaphore is responsible for the hypersensitivity reaction. Abraxane is administered through the slow infusion. It doesn't have Cremaphore, so there are no hypersensitivity reactions in response to to Abraxane, in contrast to the Taxol. So I think for cancer therapy, the current clinical examples demonstrate the utility of nanomedicines to reduce the toxicity and still provide the therapeutic efficacy. But in preclinical pipeline, there are a lot of concepts that contain targeting, like targeting to HER2 receptor on the surface of the cancer cells. Folic acid is another common small molecule used to target cancer. For colon cancer, this includes mucins and other ligands expressed on or overexpressed on the cancer cells that are missing on the normal tissues.
Okay, that'll do it. Thank you. And we have another announcement, that being that for those of you who want to get a certificate of completion of the TRACO course, you can take the final examination. We're in the process of loading that onto the website, and it should be ready by the end of the week. And basically we have one question from each of the lectures. It's a multiple guess exam, and you need to get 70% of the questions right to pass the examination. Okay, so initially we had Perez Hussain scheduled to close the course on pancreatic cancer, but he had a schedule conflict, so he spoke earlier. So I'm going to close the course talking about small cell lung cancer. And here we go. So small cell lung cancer, it's a type of lung cancer that's very deadly. It kills approximately 25,000 people in the US annually. And it's a neuroendocrine cancer. Most cancers are epithelial in nature, but this cancer is neuroendocrine. So it makes biologically active chemicals then that it can use as autocrine growth factors. So traditionally, it's treated with chemo and radiation therapy, but then relapse frequently occurs. And we mentioned before that in cancer, there's clones of cancer cells. And initially, the first clone may be responsive to chemotherapy. But then as that clone is killed, a second clone may grow out, and then it may be resistant to chemotherapy. And then that can lead to death of the patient. In any event, the small cell lung cancer is very deadly. The median survival time is less than a year. And so when you do the pathology analysis, you see that there's lots of very small cells, very prominent nuclei in the tumor. So it's very easy for the pathologist to distinguish small cell lung cancer from other types of cancer. And using the electron microscope then, you see that there's these granules in the small cell lung cancer cells, and these contain biologically active growth factors. So with small cell lung cancer, like most lung cancer, initial symptom is cough, and then you get pain in your chest. What happens is the cilia in the normal lung cells disappear as the cancer progresses. So then you can't exchange the carbon dioxide readily for oxygen, and you have shortness of breath. And in turn, that can lead to bronchitis or even worse, pneumonia. And a telltale symptom of lung cancer is when you cough, and you cough up blood. So you have bloody sputum. And so it's traditionally diagnosed with a chest X-ray, but there's other ways of detecting it as well. And a very popular way now is using spiral CT for early diagnosis of the lung cancer. So here's a chest X-ray, and you see a mass here, and this is a cancer then in the lung. You see the ribs. And using a CT scan, you see abnormalities of growth. These are tumors. And using a bronchoscopy, you can see a protuberance in the lung, in the throat. So in terms of staging of lung cancer then, you can use various techniques such as the CT scan, MRI, PET scan, radionuclide scanning, bone scan, and assaying the lymph nodes around the lung. So traditionally, if the cancer is detected early, they try to do surgery, but with small cell lung cancer, their survival time is only 6 months. And using radiotherapy, the survival time slightly increases to 10 months. but it's all goes very fast and there's death within a year. So small cell lung cancer is very responsive to chemotherapeutics, and we see a wide variety of chemotherapeutics that are used, such as the platins, carboplatin, cisplatin, and then there's various entities, such as the irinotecan, which is a topotecan analog. And then active combinations, combination chemotherapy. So you can combine cyclophosphamide, doxorubicin, and VP16, doxorubicin, vincristine, cisplatin, and there's other combinations as well. So the best thing, though, is to combine radiotherapy with the chemotherapy, and the survival then increases from 10 up to 34 months. But then there's often relapse as the second clone grows out, and after relapse, the chemotherapy is often ineffective. And the small cell lung cancer undergoes metastasis to various organs, such as the liver, the bone, the lymph nodes, and when the metastasis goes to the brain, the patient just has a few weeks to live. So we mentioned before that most lung cancer, it's associated with tobacco smoke, and you inhale various carcinogens, they get activated and mutate the DNA. But really very little is known about the origin of small cell lung cancer. Is it derived from initially endocrine Kolchitsky cells in the lung, or possibly from stem cells? We don't really know. One thing that's in the tobacco smoke is nicotine, and various carcinogens such as N and K, they can activate AKT, leading to survival of the cancer cells. So the nicotine can bind to acetylcholine receptors on the lung cancer cells, causing AKT phosphorylation. And we mentioned before that N and K forms DNA adducts, and this can lead to mutations in the lung cells. N and K also causes AKT phosphorylation. So N and K is then metabolized to N-now, and this chemical is then secreted into the urine. So N-now then represents a unique metabolite which can be measured in the urine of patients by gas chromatography. So the only way you can get N-now in your body is if the cigarette smoke is then breathed in. And then this results in an indication that you've been exposed to cigarette smoke. So, and now it's increased in non-smokers who breathe in cigarette smoke. So secondary smoke can also increase the N-now in your body. So a very unique thing of the lung cancer is in the 1980s, NCI had a program where they got biopsy specimens from patients, and then they made cell lines out of these biopsy specimens. So with lung cancer, we have hundreds of cell lines then that can be studied, and this has facilitated the research enterprise. In contrast, other tumors, you're lucky to have a dozen cell lines that you can work with. So we have hundreds of cell lines then for lung cancer study. And basically, the bone marrow aspirates were collected from patients and mononuclear cells isolated. Then the lymph node aspirates and other tumors were mechanically dissociated and cell suspensions obtained. And the cells were then put in a serum-free medium. And all the normal cells died, but the cancer cells can make their own growth factors, and hence they grew. And for the small cell lung cancers, surprisingly, they grew as floating cells. They do not adhere to traditional Petri dishes or flasks. And so for the small cell lung cancer then, we have neuroendocrine tumors. And these vesicles in the tumors, they secrete growth factors. The growth factors can then bind to receptors on the cell surface, stimulating growth. And we'll be looking especially at IGF-1. It's a tyrosine kinase receptor. And we'll be looking at the bombesin-like peptides and their G protein coupled receptors. So both of these are very important for the growth of small cell lung cancer cells. So between 1982 and 31 small cell lung cancer cell lines were established. And these cell lines, when you inject them into nude mice, tumors then rapidly form. And these small cells, they have high levels of bombesin-like peptides. They have high levels of dopa decarboxylase, which is an enzyme that's traditionally found in neurons. And they have high levels of neuron-specific enolase, which is a glycolytic enzyme that's normally also in neurons. So hence, these were then classified as neuroendocrine tumors. And over a 20-year period, over 100 small cell lung cancer cell lines were developed, as well as 100 non-small cell lung cancer cell lines. So looking at animal models, Dr. Jockalou earlier in the course talked about the AJ mouse. And here, this is a special mouse strain where you inject it with various carcinogens such as urethane, and then adenomas form on the surface of the lung. And basically what you do then is you count the number of adenomas on the lung as an index of how many tumors you've got. So for the various lipids in the cell lines, one thing that you can study is production of prostaglandins as well as leukotrienes. They're both derived from the metabolite arachidonic acid. And the cyclooxygenase enzymes in particular make prostaglandins from the arachidonic acid. So the non-steroidal anti-inflammatory drugs such as aspirin or indomethacin inhibited the growth of the lung cancer cell lines. In this case, aspirin was used. And when you added prostaglandin E2, the growth inhibitory effects of the aspirin were reversed. So this suggests that the prostaglandin E2 in particular is a mediator of the lung cancer growth. In this case, animal studies were done, and in the presence of indomethacin, the number of adenomas in the lung was reduced relative to animals that were not given any treatment. And if you looked at specimens then of the lung in the mice, and you looked for cyclooxygenase 2 enzyme by immunostaining, you found a large amount of enzyme in the bronchus. In this case, the color of the immunoreactivity is brown, whereas the purple is just a counterstain of the tissue. And you see lots of immunoreactivity in the bronchus, some in the bronchioli, as well as the alveoli of the lung. And here we see intense staining in the bronchial epithelial cells, moderate staining in the muscle, but not the cartilage. In the bronchioles, there's moderate staining in epithelial cells. In the alveoli, moderate staining in type 2 cells. And in the adenoma, there's also scattered cellular staining. So there's two types of cyclooxygenase enzymes, the cyclooxygenase 1, which is constitutive, and then the cyclooxygenase 2, which is inducible. And here in this experiment, we added epidermal growth factor to cell lines. We saw that the cyclooxygenase 2 mRNA increased. So it's really thought that in lung cancer, the cyclooxygenase 2 enzyme is very important for proliferation. And the cyclooxygenase 2 enzyme will then make prostaglandin E2. And if you have cell lines and you add prostaglandin E2 to the cell lines, you get phosphorylation or activation of the EGF receptor. And this AH is a G protein coupled receptor antagonist that blocks the type 2 receptor on the lung cancer cells. So prostaglandin E2, not only did it phosphorylate the EGF receptor, it also caused ERK phosphorylation in a dose-dependent manner. And then the phosphorylated ERK can go into the nucleus and turn on various oncogenes. When various genes are activated at AP1 sites, then ultimately VEGF can be expressed. VEGF is an angiogenic factor, so here we see we get a doubling of VEGF when we add prostaglandin E2, and EGF will also increase the VEGF mRNA. And here's a little cartoon summarizing what we think is going on. That being that the EGF receptor will activate the COX-2, increase its expression, leading to increased PGE2 production. The PGE2 binds to the G protein coupled receptor, the EP2 receptor. The EP2 receptor then can cause transactivation of the EGF receptor, phosphorylation. So this is basically an autocrine growth cycle then. It's started by the EGF receptor, and then ultimately transforming growth factor alpha gets released, which further activates the EGF receptor. And once the EP2 receptor is activated, it can also increase cyclic AMP, causing PKA activation, Krebs phosphorylation, and an increase in VEGF expression. So the prostaglandin E2, it's not only a growth factor stimulating the EGF receptor, it's also an angiogenic factor, increasing VEGF expression. So in terms of COX inhibitors, the nonsteroidal anti-inflammatory drugs can inhibit both COX-1 and COX-2, but when you take aspirin, side effects can occur, such as stomach ulcers. So subsequently, a great effort was made into making selective COX-2 inhibitors, and the one that's traditionally used is celecoxib. And the goal of celecoxib then is to block the cyclooxygenase 2, but have minimal side effects in patients. And so there's lots of clinical trials going on using celecoxib. And there's phase 2 studies studying the effects of celecoxib in heavy smokers. There's also phase 2 studies studying the effects of celecoxib in the presence of chemotherapeutic drugs such as paclitaxel and carboplatin. So in small cell lung cancer then, we have several things going on, such as there's RB inactivation in 90% of the patients. We mentioned P53. It's a tumor suppressor gene. It gets inactivated in about 90% of the small cell lung cancer patients. Another thing that's inactivated is FIT in about 3/4 of the patients. And then BCL2, which causes survival of cancer cells, is overexpressed in about 85% of the small cell lung cancer patients. So again, in lung cancer, we mentioned there's no one thing that causes it, but since it takes decades for lung cancer to form, several things lead to the lung cancer proliferation. So with P53, we mentioned previously, it causes G1 to S phase checkpoint inhibition in most of the cells, subsequently driving programmed cell death or apoptosis after DNA gets damaged. So if the P53 gets mutated, then the cancer cells will readily proliferate. For the retinoblastoma gene, there are mutations such as truncations, deletions, nonsense mutations, and splicing abnormalities that can occur in the lung cancer patients. Also, the wild type allele is frequently lost in small cell lung cancer, and subsequently the RB protein is pretty much inactivated in 90% of the small cell lung cancer patients. The FIT gene is fragile histidine triad. It's present on chromosome 3p14, and loss of the FIT protein is associated with smoking, and there's a lot of investigation going on to see if FIT is a tumor suppressor gene associated with apoptosis. So for the BCL2, it's overexpressed in about 85% of the small cell lung cancer patients. The BCL2 suppresses apoptosis and responses to chemotherapy and radiation therapy. So there's lots of clinical trials being done, basically trying to stop the BCL2. So in terms of molecular abnormalities, there's loss of chromosomes in 3p, 4p, 4q, 5q, 8q, 9p, 10q, 13q, 17p, and 22q. We'll also look at microsatellite instabilities, MYC overexpression. MYC is an oncogene. Tyrosine kinase receptors such as the c-KIT overexpression, and we'll close looking at the bombesin GRP receptor, the G protein coupled receptor. So 3p dilution is very dramatic in the chromosome of small cell lung cancer patients, and 3p is a very early event. And subsequently, 5q, 13q, and 17p deletions will occur. The p53 is on 17p. Microsatellite alterations. In lung cancer, there's a laddering of short tandem DNA repeat sequences at multiple loci. This laddering may result from mutations in DNA mismatch repair enzymes. This microsatellite instability may be useful for the early diagnosis of lung cancer using sputum, bronchial washings, or blood. MYC is an oncogene, and NMYC and LMYC are amplified in small cell lung cancer. MYC then can heterodimerize with MAX and facilitate cell cycle progression. Another thing that happens is the LKB1, it's a serine threonine kinase that's inactivated in about half of the small cell lung cancer patients. So LKB1 causes phosphorylation of AMP activated protein kinase, resulting in tumor growth. And the IGF-1 receptor, it's a tyrosine kinase receptor. In contrast to the EGF receptor, it binds insulin-like growth factor 1 with high affinity. And it's actually a dimer where the IGF-1 binds to the alpha subunit. The tyrosine kinase activity is then on the beta subunit. So when the alpha subunit binds IGF-1, it causes a conformation change in the beta subunit, causing protein phosphorylation. And downstream events include activation of MAP kinase, growth of CFOS, and increased expression of CFOS, and then growth of the lung cancer cells. So here's the IGF-1 receptor, and we got a monoclonal antibody, alpha IR3, which binds to the IGF-1 receptor and recognizes the 90 kilodalton subunit. So this antibody was then tested in nude mice bearing small cell lung cancer tumors. You see the tumors exponentially grow, and in the presence of the alpha IR3, the tumor growth was greatly slowed. So the IGF-1 then enhances survival of the small cell lung cancer cells, and it phosphorylates the PI3 kinase, leading to phosphorylation of the AKT and increased survival. So the PI3 kinase inhibitor, LY294002, inhibits the phosphorylation of AKT caused by IGF-1. And here we see the tyrosine kinase receptors activating the PI3 kinase, which then causes the phosphorylation of the AKT and increased expression of the BCL2, inhibiting apoptosis of the lung cancer cells. So another tyrosine kinase receptor that's present on the small cell lung cancer cells is c-KIT, and scatter factor binds to c-KIT, and the c-KIT is often overexpressed in the IGF receptor in the small cell lung cancer cells, and the c-KIT can be inhibited by Gleevec. So the c-KIT, it's a 976 amino acid protein. 520 amino acids are on the extracellular face. There's a single 23 amino acid transmembrane domain, and then 433 amino acids are on the inside that contain the tyrosine kinase activity. So ATP binds to the tyrosine kinase domain, and various proteins then are phosphorylated on tyrosine amino acid residues. So finally we get to the GRP receptor, and I published in 1980 that the small cell lung cancer cells had very high levels of bombesin-like peptides. And we then elicited an antibody against the GRP. And in nude mice, you got large tumors, but animals given the antibody, the tumor size was greatly reduced. So subsequently, clinical trials were tried with this antibody, but it only worked in 1 out of 13 patients. So subsequent efforts were discontinued. But then the field sort of switched, and antagonists were developed against the GRP receptor. And so you see the GRP receptor, it's got 384 amino acid residues, crosses the plasma membrane 7 times, and the GRP binds on the extracellular side. And what happens is this third cytosolic loop will then cause phosphatidylinositol turnover. So when GRP binds to the receptor, phosphatidylinositol gets metabolized. And a very curious thing is with men now, the cancer incidence in the lung is rapidly decreasing. With women, it's sort of leveling off. And one interesting thing about the GRP receptor is it's on the X chromosome. And so subsequently, the females have a double dose of GRP receptor relative to males. And it remains to be determined if this is why women are more susceptible to getting lung cancer than men. So in this case, we're adding GRP to small cell lung cancer cells that are loaded with dye, and we're watching the cytosolic calcium go up as a function of time. And what happens is when the phosphatidylinositol gets metabolized, IP3 is one of the resulting products, and then this will cause release of calcium from intracellular organelles. So when we add the GRP, after 30 seconds, we see many of the cells are starting to light up. It peaks after about a minute, and then it readily declines with time. So the increase in cytosolic calcium is very transient. This is a different way of looking at it graphically. We add the GRP, and the calcium increases, and it's a transient increase. And then we identified antagonists that block the receptor, and they block the increase in calcium. So these antagonists, they're small molecules, and the one that blocks the GRP receptors is PD176252. And another thing that the GRP receptor can do is it can cause transactivation of the EGF receptor. We saw previously that prostaglandin E2 will do this, but also GRP will also do this. And the increase in EGF receptor tyrosine phosphorylation is very quick. It occurs within a minute. And then the antagonist, the PD176252, will block the increase. And when the EGF receptor gets tyrosine phosphorylated, events downstream include ERK activation, and here we see that bombazine causes the ERK activation, and the antagonist will block that as well. And in terms of growth, one of the things that we mentioned previously is the gefitinib is used in patients with mutated EGF receptor. So one goal is then to increase the potency of gefitinib in patients with wild-type receptor. And we find that when we add this antagonist, the PD176252, the gefitinib dose-response curve starts shifting to the left. And so the PD176252 increases the potency of gefitinib by over an order of magnitude. So it's possible then that this bombazine receptor antagonist, in combination with gefitinib, may be used to inhibit the lung cancer growth. So in summary then, we see that small cell lung cancer, it's a neuroendocrine tumor. It initially responds to chemotherapy, but relapse then occurs. There's multiple clinical trials in progress to improve the treatment of small cell lung cancer patients. And then we want to close by focusing on smoking. So 85% of the lung cancer cases, the patients are smokers. And so nicotine is what comes out of the cigarette smoke that makes it very addictive to various people. So in terms of getting people to stop smoking, one of the things that they do is there's lots of nicotine replacement therapy now. So this includes the gum, such as Nicorette. You can use a patch, which is the Nicoderm. There's a nasal spray, Nicotrol, and now there's even a nicotine lozenge. And each of these delivers on the order of 10 mg of nicotine per day to patients. And there's also some pills that are used, such as bupropion, which is an antidepressant. But lately, the thing that's very in vogue is using Santix. And this basically reduces the smoking urge and withdrawal symptom. So there's lots of things that now you can take to get over the need to have nicotine. And then the question is, will that help you stop smoking? And in many cases it does, but this is a very difficult thing to get people to stop smoking. Basically, if you start smoking when you're a teenager, it's very hard to give up. And people will often smoke for 10, 20, even 30 years. But if adults start smoking later, such as when you go to college, it's fairly easy to give up. And these people, they tend to smoke for just a few years, and then they stop. So what the U.S. has done is they tax the heck out of tobacco. And as a result, the teenagers can't afford it, and the number of teenagers smoking has gone down dramatically. But for people who are still smoking, cessation can be achieved with the use of medications in healthcare professionals. And early failure is a normal part of trying to stop smoking. So people, when they try to stop smoking, oftentimes it'll work for a short while and then they'll go back. But then a year later they may try again to stop smoking. And again, it doesn't work. But eventually, it does work, and people will stop smoking. And smoking cigarettes certainly leads to nicotine addiction, but ultimately, the way most smokers quit is they just quit cold turkey after a gradual reduction in the number of cigarettes that they utilize each day. So the health benefits of stopping to smoke: within 20 minutes, blood pressure starts to decrease, heart rate decreases. Within half a day, carbon monoxide levels in the blood return to normal. Within 2 days, the sense of smell and taste returns. Within 9 months, there's a decrease in cough and a shortness of breath. Within 10 years, the risk of stroke is normal, and the risk of dying from lung cancer is reduced by 50%. So now in this country, we still have 45 million smokers. The good news is only 1 in 6 will actually die from lung cancer. We don't know why all the smokers won't die from lung cancer. Many of them die from other things, though, such as heart disease. But now in the US, we have 45 million ex-smokers. And that's a very, very good thing, because after 10 years, the risk of getting lung cancer is dramatically reduced. And here's some references. And that's the end of the TRACO course. Are there any questions? Okay, well, thank you all for attending, and I wish you all a happy holiday season. And again, we'll have the exam up soon if you want to get a certificate of completion from the course. Thank you.